Hi, everyone. Good afternoon. We're going to go ahead and get started. My name is Mirat Shah, and I'm a medical oncologist specializing in breast and gynecologic malignancies at the U.S. Food and Drug Administration. And on behalf of myself, as well as my co-moderator, Dr. Patricia LoRusso, and the other speakers and panelists that are part of our session, I'd like to thank the session organizers for inviting us to give this clinical workshop on dosage optimization, a topic that is important and extremely timely. I will walk everybody through the program for the next hour and 45 minutes. Then I will invite my co-panelists to introduce themselves, and then we'll get started with the talks. So the session is divided into two parts. The first part will, the first part will focus on the topic of dosage optimization. The first talk will introduce dosage optimization and why this should be such a big focus right now. The second talk will go into the nuts and bolts of dosage optimization. After that, we will present the related concept of regimen optimization and finish with a patient perspective on dosage optimization. Following those four talks, which all relate to one another, we will pause for questions and panel discussion. Then we will transition topics a bit and talk about crossover, an extremely important topic for oncology clinical trials. That will be followed also by a short Q&A and panel discussion, and then we'll conclude the session. So with that, I will start over here to my left and invite everybody to introduce themselves.
Hello, everyone. My name is Stacy. I'm a clinical oncology pharmacist by training, and I work in the FDA at the Office of Clinical Pharmacology.
Hello, everyone. I'm Maués. I'm a patient advocate and the lead of the Patient-Centered Dosing Initiative.
Hello, my name is Ignatiadis. I'm a medical oncologist working in Brussels at the Jules Bordet Institut.
Mallory Fierro. I'm a supervisor in the Office of Biostatistics, and my team support the breast cancer teams at FDA.
Hi, and I'm LoRusso. I'm a medical oncologist at Yale University, Yale Cancer Center, and I run the early phase clinical trials program there.
Thanks, everyone. So I wanted to let the audience know that by common agreement, the panel has agreed to refer to each other by first name to keep this conversation informal and to invite all of you to feel very comfortable asking all of us questions. A few other housekeeping notes. My co-panelists and I will mention some case examples within the context of our presentations. The purpose of those case examples is really to highlight important concepts, but this is not meant to be a session that goes into the nitty-gritty of each of those cases. And then finally, we want this session to be interactive. We want all of you to learn from us, but we also really, really want to learn from all of you. So really welcome everybody to submit questions online and come up to the microphone during the Q&A portions. Thanks, and I think with that, we can get started with our first talk.
Thank you. Thank you for inviting me to talk, and also thank you all for being here at this session. Obviously, dose optimization is pretty near and dear to my heart because I've been working on early phase clinical trials for over 35 years. And, back in the heyday when I first started, older than most of you in the audience probably, we had 5-fluorouracil, and in the lab we were initially developing cytotoxic agents. So drugs have morphed significantly based on the biology of the disease that we now understand more, and also being able to have the novel tools that we have to help develop these drugs. I do not work at the FDA, and I don't represent the FDA. These are my disclosures. And I guess that's my talk. Oh, here, okay. There you go. Sorry, I have a lot of studies that I do. So the first question I think we need to ask is, are we getting the dose right? And I think after many, many years of probably not getting the dose right, and I'll show you some examples, I think we're heading in the right trajectory thanks to some of the guidances that have recently been developed by the FDA. But when we were dealing with cytotoxic agents, there was a very narrow therapeutic window between efficacy and toxicity. But that changed with the development of many of these other drugs, including molecular targeted agents. This study was from 2016, but I think it's a really good example. As early as 2016, we were concerned about whether or not with these drugs, and we were developing molecular targeted agents at that time, were we really getting the drug right, or were we overdosing our patients? And so this study looked at 90 oncology drugs that received FDA approval between January of 2002 and February of 2015, 34 of which were MTAs, 28 small molecules, 6 targeted antibodies. And the conclusion from that study and that retrospective review was that 45% of patients on small molecules required dose modifications due to drug-related toxicity in subsequent phase 3 trials. And as you can see, 48% were interruptions, 42% were dose reductions. And this is really important because as we're trying to develop combination strategies, and with most MTAs, especially in the metastatic setting, combinations are probably are very important, but are very, very challenging because of overlap and toxicity. So it's important to look at ranges, I believe, in trying to get the dose right and identifying whether or not that dose is right for all tumor types and in what settings. So what they did identify, though, in this study is that 25% of small molecule MTAs that had phase 1 trials which recommended a phase 2 dose below the maximum tolerated dose based on kinetics and dynamics had fewer dose modifications in subsequent phase 3 registration trials. And this is, where we were, even at the time that that retrospective study was done. Because we had been focused for so many years on narrow therapeutic window, we failed to recognize that with a wider therapeutic window, we had to really look at the dose. And as Stacy will probably be talking about in the next several minutes, trial design was becoming more important than ever. Obviously, the narrow therapeutic window was with cytotoxic drugs, but then based on the biology of the disease and science, we developed a myriad of other drugs, not only targeted, but monoclonals, bispecifics, et cetera. And each of these has different pharmacokinetic and pharmacodynamic profiles, and we need to take that into consideration as we're advancing the drug beyond phase 1 trials. And so the new paradigm, as you can see with MTAs, is that there's a significant differential between efficacy and toxicity. And with some of your bispecifics, there's actually, as you can see, a bell-shaped curve beyond where you need to go with the dose versus beyond that, you may be, especially if it's an agonist, beyond that dose, you may be causing more harm than good, primarily in efficacy, but also in toxicity as well. So with the development of these novel agents, as you can see, we went from virtually no therapeutic window to a wider therapeutic window, and with many of the small monoclonal antibodies, even a greater therapeutic window. And so what did that mean in terms of how we were going to, or what we needed to do in terms of where we were with trial designs versus where we needed to go, and what we needed to look at within the context of those trials, not just revving up to the maximum tolerated dose, but identifying additional components. So this was the classic 3+3 design, the modified Fibonacci or whatever, the classic 3+3 design, where you treated 3 patients, you went up, you continued up until you got to a dose that was too high, backed 1 or 2 dose levels until you got a more relevant or more tolerable dose, expanded to 6 patients, and then you might expand with a certain number of patients in select windows based on what tumor types you might want to look at. But this is where we are and where we need to continuously focus. Sure, we still need to do dose escalation because we still need to prove that the safety of that the drug is safe. But as we're going through, and many times still in 2024, we're still pushing drugs to the maximum tolerated dose because depending on the drug class, it may be the only opportunity that you have to see what the maximum dose is that you can deliver. And you're looking for dose-limiting toxicities as well as what the maximum administered versus maximum tolerated dose is. But hopefully, because biology is driving these new novel therapies, we identify the pharmacodynamic effects relatively early in the dose escalation schema. And after that pharmacodynamic checkpoint, we may start seeing, we're hoping to see responses. And hopefully we're going to see responses at and above that PD endpoint. And in multiple cohorts. And we then eventually, as we're escalating, we identify that as the maximum as the biologically effective dose. And below that, for many reasons, based on efficacy as well as biologic or pharmacodynamic endpoints, we have a subtherapeutic range. Based on the efficacy that we see as we're escalating, a therapeutic range. But where is the recommended phase 2 dose? Is it still the maximum tolerated dose? And I think that's why Project Optimus was really pushed forward, because it doesn't have to, and it may not even be the maximum tolerated dose, because as you push the drug to MTD and expanded MTD and have several dose reductions in your subsequent patients, especially when it leads to post-approval, you don't really know what the efficacy is relative to all those reductions. And so pushing at the MTD may actually do the drug significant injustice, but more importantly, the patients that will suffer toxicities as a result of that. So what I believe in what we're doing in 2024 and starting to really rev up is looking at the totality of data needed for dose optimization. What are the key ingredients for success? The drug and the mechanism of action, the drug class. We know more if it's not first in class, but depending on the differential in the chemistry of the compound, it may still have significantly unique properties. The patient population, the histology selection, as well as molecular biomarker selection. Because what we're seeing, and I was just on a safety call this morning, looking at what the recommended doses that we were going to be moving forward, depending on the histologic subtype and the expansion cohorts that we had in our trial, in our phase 1 trial, we identified a different dose predicated on the two histologies. What are the toxicities? Not only acute, but more importantly with these drugs, we're seeing a lot of chronic toxicities, and how do we integrate those? Pharmacokinetics is obviously very important. We need to know what the dose-PK effect relationships are, as well as the biologically relevant concentrations that we need to achieve based on a totality of data, preclinical as well as clinical. Pharmacodynamics, are we hitting the target? We need to know that, and what is the proof of mechanism for that drug? And then finally, clinical activities that support evidence, such as things like tumor markers, as well as ctDNA molecular responses. Our patients in phase 1 are heavily pretreated. We may have to rely on some of these other clinical endpoints and biomarkers to help guide us to dosing and subsequent trials. But we need the totality of the orthogonal data to make informed go, no-go decisions to increase the odds of success in drug development. Biomarkers are extremely important. As these drugs are being developed based on the biology of the disease, if we don't develop biomarkers early on and test them in proof of concept with early phase trials, we may not really understand the magnitude. And if we don't develop biomarkers, many times it's very difficult, especially predictive biomarkers for success. We've seen randomized phase 3 trials where we now have drugs in the neoadjuvant setting, but the biomarker strategy was not there. And as a result, many patients may be treated with drugs that can cause toxicity, for which we may not be getting a therapeutic benefit. There are many different types of biomarkers that we use. We use next-gen sequencing, hopefully things that in the future that'll assess functionality as well as quantities such as phosphoproteomics. We can enrich, and we should enrich dose escalation population with the predictive biomarker, which includes a priori provisions in the protocol to add additional patients if they have that biomarker, as well as backfill cohorts. The end result is what we need to do is try and make every patient count and make the drug count in every patient, even in early phase clinical trials. And we need to mandate expansion cohorts, especially with molecularly driven tumors. The advantages, they allow early clinical testing and validation of these assays, permit early hypothesis testing and generating studies, and optimize the chance of early anti-tumor signals, decreasing, hopefully, the number of patients receiving ineffective treatments and avoid drug development fatigue for the drug, and may avoid late drug attrition and subsequently, in the long run, reduce costs. The caveat is combinations may still be needed to optimize anti-tumor activity. So I just want to show you a couple examples that the whole breast cancer community is aware of as to why Project Optimus and dose optimization is very important. So let's look at fulvestrant. Obviously, poor solubility. It needed to be administered via IM injections because of limits in volume and dose. It was originally developed as a drug for non-malignant disease. In Japan, it was being tested at 25 milligrams, 50 milligrams for uterine fibroids and endometriosis. And then in cancer trials, they moved forward with an initial dose of 250 milligrams, day one every 28 days. And as you can see, there were 3 large phase 3 trials with different hormonal interventions that comparing different hormonal interventions to fulvestrant. But there was much debate over optimal dosing, which led to further investigation, because at 250 milligrams, there were some issues, and it did not have any significant toxicities or minimal toxicities at that dose. So they went forward with a biomarker-driven trial, the newest randomized phase 2 trial, where they looked at 500 milligrams, which was also safe, and 250 milligrams. What they identified was 500 milligrams was a dose that was associated with significant increased downregulation of estrogen receptor, which led to the confirmed phase 3 trial, which subsequently not only identified that 500 was superior to 250, but because of the formulation of the drug, it takes significant doses, if you're only giving it once every 28 days, to get to a steady state. So you were doing a huge disservice to the patients. And as a result, they identified that day 1, day 14 for cycle 1 was going to enhance the ability to get the exposure to a steady enough state to make it a meaningful dose. But for many, many thousands of patients, until that COMPARE trial was done, patients were underdosed, minimizing the true efficacy that they could have obtained from that drug. I was involved in capecitabine, and I can't believe we did trials like that back then. But as you can see, this was the phase 1 trial of capecitabine. We went all the way up to 3,500 milligrams per meter squared, 406 DLTs, dropped it down to 3,000, 2 out of 6 DLTs, and the 2,500 milligram per meter squared dose advanced to subsequent phase 2 registration trial, but it had 1 out of 9 DLTs. We had 135 patients. There were methods in the trial where we accounted for dose de-escalation based on toxicity, but the majority of patients, many, many patients, had to be de-escalated. But nonetheless, the 2,500 milligram per meter squared divided at 1250 BID was advanced in this trial. These patients were treated at that dose despite de-escalation. As you can see, there was about a 20% response rate, 30% stable disease rate. The drug became FDA approved. However, capecitabine is the classic example of why Project Renewal needed to be developed by the FDA as a guidance, because very few of you, I'm sure in the audience, use 1250 milligram per meter squared BID daily times 14. It's just too toxic. Patients can't tolerate it. And as a result, Project Renewal was specifically stated, and this was the first drug to get approved to go through Project Renewal, that it updated the labeling for the capecitabine tablets through the Oncology Center of Excellence initiative, and it gave altered recommendations of 1,000 or 1,250 milligrams per meter squared BID. So what is Project Optimus in my eyes? I'm going to show you what I think I don't work for the FDA, but this is a drug that's moved on, I believe, to phase 3 trials that is being developed by AstraZeneca. And I look at Project Optimus akin to the pharmacologic audit trail that was actually developed by Paul Workman in 2010. And I think it's an updated and new rendition. And I look at Project Optimus as bringing a drug through, requiring checkpoints at 5 pillars for drug approval or drug development. So let's look at AZD5305. It's a PARP1 selective agent. As you can see, the phase 1 trial was done starting at 10 milligrams through 140 milligrams. It was obviously done in patients that had those alterations. And at dose expansion, because they saw activity in other PK/PD markers early on, they looked at 3 doses. In expansion, they looked at 20, 60, and 90 milligrams daily. So what about non-clinical data? As you can see here, phenomenal in vivo activity. I apologize for the size. Significantly better PARylation. It was PARP1 selective. Significantly better PARP1 trapping. Again, it was PARP1 selective, unlike any of the first-generation PARP inhibitors. And preclinically in models, it was devoid of the same toxicity, both peripherally as well as blood erythrocytes, that the first-generation agents had. Pharmacokinetics, this drug had beautiful linear pharmacokinetics, and the therapeutic effective concentration at the first dose of 10 milligrams superseded the TEC of any of the first-generation agents, and up through 140, the TEC was increased by 137-fold over those first-generation agents. Very good exposure. In terms of pharmacodynamics, ctDNA predicted responders over non-responders, and PARylation inhibition following a single oral dose of 5305 confirmed robust target engagement. In terms of safety, the phase 1 trial, there were no dose, I think there was 1 patient in the entire phase 1 trial that required a dose reduction. And they did logistic regression analyses while they were doing the 20, 60, and 90 milligram dose and identified that 90 milligrams might be a little too high in terms of development of anemia. So they backed off on the 20 and went to the 20 and 60 milligram dose. You saw significant activity with this drug, not only in terms of waterfall, but also swimmer's plots and spider plots, showing that indeed this drug was safe. 60 milligrams had slightly, as you can see, to, I don't know, it's the waterfall plot in the bottom, my bottom right, had somewhat better activity in terms of the spider plots and the waterfall plots than 20 milligrams. Both were safe and easy to administer, and as a result, AZD identified 60 milligrams as the optimal dose to advance into phase 3 trials. As a phase 1 or early phase clinical trialist, I don't think there's going to be one set recipe for every drug. I think that we know there's no need to think outside the box, but simply realize that there is no box, that we have to take the totality of what we're dealing with, including the biology of the drug, in order to advance these drugs into early phase clinical trials. And I can tell you, after doing this for over 35 years, I'm more excited about doing early phase trials in 2024 than I think I was when I just started off as an assistant professor, because of the novelty of designs and the biology behind what we're doing and the advancements that we've made in clinical cancer research. So finally, one size does not fit all. You need to take it all into consideration. And it's important, I think, in conclusion, to get the dose as right as possible prior to phase 3 clinical trials. Because if you have too many de-escalations, you really don't know what the true efficacy would be when you get into the standard of care. Ramifications of too much or too little drug could have significant consequences on patient care. I think Project Optimus is amazing because it focuses on the totality of data. It brings the science into the clinical trial of the drug for improved dose decision-making, take into account all the available data, minimizing toxicity, enhancing efficacy, and improving outcome for our patients. Thank you.
Good afternoon. Thank you to the organizers for asking me to participate in this important session, and to Dr. LoRusso or Pat for providing an overview of the current need for dosage optimization in breast cancer. I look forward to discussing with you the basic principles driving dosage optimization for oncology products and highlighting some of the unique challenges associated with dosage optimization for oncology products developed by breast cancer and other cancers. This slide lists my disclaimers. Today, I will briefly address the 3 questions listed here. Why is there a renewed interest in dosage optimization? How is the focus on dosage optimization changing drug development and clinical trial design? And what resources are available to navigate this new drug development paradigm? I will start by addressing the first question. In the past 20 years, we have witnessed a dramatic change in the types of oncology drugs being developed. These graphs clearly show the shift from traditional chemotherapy agents to targeted therapies. Targeted therapies, as listed earlier today, include small molecule drugs, antibody-drug conjugates, and bispecific antibodies. The number of targeted therapies has dramatically changed the treatment landscape for many cancers, including breast cancer. Next, I would like to discuss why this remarkable trend is forcing a change in drug development and clinical trial design. Key differences between cytotoxic chemotherapies and targeted therapies, as listed here, are necessitating a change in how we design clinical trials to identify and select the recommended phase 2 dose for registration trial and ultimately the drug labeling. Classic dose-finding trials have focused on identifying the maximally tolerated dose, or MTD, and identifying the dose-limiting toxicities observed in about 1 month following the first dose. Both rule-based and model-based designs have been implemented to guide dose escalation in these trials. Despite the trends we reviewed on the previous slide, this paradigm has changed little, with the objectives still largely remaining to identify the MTD. As a community of healthcare providers, scientists, and regulators, we recognize that this paradigm might not be the best to identify an optimized dose for the targeted therapies. For targeted therapies, differences in exposure response and dose response mean doses below the MTD may have similar efficacy to that of the MTD, but with fewer toxicities. Additionally, the MTD may never be reached in certain situations. For targeted therapies, we also need to account for the longer treatment duration, as some cancers require chronic, long-term treatment to retain benefit. Given these inherent differences, alternative trial designs are warranted to identify optimized dosages for targeted therapies that balance safety, tolerability, and efficacy. But the biggest push to change our current practices is patients. Interactions with patients, caregivers, and advocates clearly show that patients want alternative dosages to that of the approved recommended dosage. Patients express desire to tolerate a recommended dose for long periods of time to retain their clinical effectiveness. Concerns evolve around dosages that lead to adverse reactions that negatively impact tolerability and their quality of life, and little to no information on the clinical effectiveness of alternative dosages or schedules. So how do we go about finding an optimized dosage for targeted therapies? After the initial dose escalation portion shown in step 1, most trials include dose expansion portion. The expansion portion historically evaluated a single dose in 1 or more patient populations. Some trials may also incorporate a second dose escalation portion to evaluate various combination therapies. These traditional dose escalation portions of the first-in-human trial are typically conducted in a limited number of patients at a limited number of dosages, and the dose selected for expansion portion is often the MTD. Therefore, this initial dose escalation portion provides very limited clinical information and cannot adequately inform the dose selected for subsequent trials. FDA encourages creative approaches to further explore the safety, tolerability, activity, and exposure of more than 1 dose before moving into step 3. These creative approaches should consider all non-clinical and clinical data, with some common data being considered in this new paradigm shown in the red box. These data may include the relevant pharmacokinetics, pharmacodynamics, activity, efficacy, safety, and tolerability data at each step of the development. FDA recommends that additional safety information, not just the dose-limiting toxicities, be evaluated. This may include low-grade symptomatic toxicities, dose modification frequencies, and patient-generated data for treatment-related symptoms, also known as PROs. This holistic approach includes identifying a target dosage range early and then further evaluating several dosages in several subsequent trials before arriving in step 3. Lastly, this approach also includes characterizing the dose and exposure-response relationships for efficacy and toxicity, starting with the early emerging clinical data and incorporating new data as it becomes available. The exposure-response and dose-response relationships can also help us understand the impact of various intrinsic and extrinsic factors, including food and concomitant medications and various patient factors on the dose. To facilitate identifying the optimized dose to be further evaluated in clinical trials, FDA advocates that multiple dosages be compared early in drug development, and these dosages be selected based on all relevant non-clinical and clinical data. Backfilling dose level cohorts may be a means to gather additional data at multiple dosages in the dose finding trial. FDA also supports a randomized comparison of 2 or more dosages. This randomized comparison may be incorporated in the first-in-human trial or as a standalone trial. Multiple unique and innovative trial designs are plausible. When feasible, FDA recommends randomization to account for known and unknown variables that impact outcomes. This randomized dosage comparison does not need to be statistically powered for superiority, but these comparisons should be sized to allow for sufficient assessment of activity, safety, and tolerability. However, FDA recognizes that given some populations may be generally small and outcomes may be very poor, a randomized comparison may not always be feasible. Therefore, sponsors are encouraged to discuss their dosage optimization plan with FDA early, and I will provide some resources later in the presentation today. Depicted here is one example of what a randomized dosage optimization trial could look like before approval. This trial compared 2 dosages of trastuzumab deruxtecan, a HER2-directed antibody and topoisomerase I inhibitor conjugate, for patients with non-small cell lung cancer. The randomized comparison evaluated the activity and key safety findings and supported selection of a dose of 5.4 milligrams per kilogram for further evaluation. The dose was the approved recommended dose for patients with breast cancer and became the approved recommended dosage for patients with non-small cell lung cancer. Although the approval for breast cancer superseded that of the non-small cell lung cancer, this example demonstrates how a randomized dosage comparison can be designed and conducted before an approval, and that it might just be plausible to conduct similar trials for new oncology drugs for breast cancer in the future. The goal of dosage optimization before approval is to identify a dosage that preserves clinical benefit with improved tolerability and safety. But is it possible to conduct dosage optimization post-marketing? To address this question, let's briefly look at the development of Inavo. The primary evidence of efficacy and safety came from the trial Inavo 120 shown here. Only 1 dose of Inavo, 9 milligrams at the MTD, was evaluated in this trial. Although the proposed recommended dose was determined to be approvable, this dosage was not considered to be optimized for the reasons listed here. Briefly, multiple common AEs were observed at higher rates in patients who received Inavo in combination and at higher incidence than those in the control arm. In addition, about 70% of patients who received the combination had at least 1 dosage interruption, and about 84% of these dosage interruptions were due to adverse reactions. These results suggest that the proposed dosing regimen for Inavo in this combination is poorly tolerated. Other concerns raised during the review included that the ER analysis, or exposure response analysis, identified a higher probability of several key safety endpoints and dosage modifications due to adverse reactions with a higher concentration of Inavo, and the limited clinical data was available for Inavo at other dosages as a single agent or in other combinations that were being explored. The exposure response for efficacy, unfortunately, was inconclusive because the dosage was limited to a single dose in our studies. FDA therefore required a post-marketing requirement to evaluate a lower dose in the same patient population. This is great news, right? No. It can be very challenging to conduct dosage optimization post-approval. Our research shows that about 15% of approved new drugs were issued a post-marketing requirement to further evaluate the dosage for a number of reasons. And it took about 6.5 years to fulfill or release the post-marketing requirement. That's a very long time to resolve dosing issues and for patients to be underdosed or overdosed during this period of time. Therefore, FDA strongly encourages in optimizing the dosing regimen before approval. Taking a holistic approach and looking at emerging data as it becomes available emphasizes optimizing the dosage before approval. Dosage optimization allows us to improve tolerability and adherence, reduce dosage modifications and discontinuations, and potentially increasing the likelihood of response to treatment. If a patient cannot continue an effective dose, given adverse reactions, the likelihood of clinical benefit will decline. Studying multiple dosages in an adequate number of patients for a sufficient duration can also help us gain an earlier understanding of the dose and exposure response relationships and allow for more rapid development of subsequent therapies such as those listed here. FDA strongly believes that thoughtful dose selection that evaluates all relevant available data as it emerges will hopefully avoid the challenges associated with conducting the dosage optimization trial post-approval as demonstrated by our research. Alternative creative approaches to dosage optimization can also help address some key questions or concerns being raised in this era of new targeted therapies and can be applied to drug development for novel therapies for patients with earlier metastatic breast cancer. Some of the key questions are listed here, but this list is far from complete. To jumpstart dosage optimization for oncology products, the Oncology Center of Excellence initiated Project Optimus about 4 years ago. Its purpose is to reform dosage optimization and dose selection paradigm for oncology drug development. The Project Optimus team includes individuals that represent a myriad of disciplines at the FDA. The specific goals include communicating expectations to stakeholders for dose finding and dosage optimization, providing opportunities for sponsors to meet with review divisions early in development to discuss dosage optimization, and developing strategies that evaluates all available relevant data to identify dosages to be further studied in clinical trials. The link provided here takes you to the FDA's external site that houses a fuller summary of the project. As part of Project Optimus and other ongoing FDA initiatives, FDA published a final guidance on dosage optimization earlier this year. The guidance provides recommendations to assist sponsors in identifying the optimal dosages for oncology drugs with an emphasis on identifying an optimized dose during clinical development and prior to submitting a marketing application. Some of the general principles listed in the guidance are summarized here. These principles also apply to drugs that are being developed under expedited programs. The Oncology Dosing Toolkit is another resource available to help collate data that can be used to inform dosage selection. These key questions mirror the pillars of dosage optimization highlighted by Pat earlier this afternoon. In summary, there is a renewed interest in dosage optimization sparked by the transition to targeted therapies. Dosage optimization requires a patient-centric, holistic approach that looks at all available, relevant, and emerging non-clinical and clinical data throughout development. Novel trial designs and quantitative approaches with alternative endpoints analyses can support our exploration to fully understand the dose and exposure response relationships for key endpoints, including efficacy, safety, and tolerability, and identify an optimized dosage for further evaluation in trials are included in labeling. Unique challenges for patients with breast cancer will require a multidisciplinary approach with multiple stakeholders to determine the best means to address these challenges and meet the goals of improving patient tolerability and outcomes. With that, I want to thank these individuals for their support.
Good afternoon again. So far, as part of this clinical workshop on dosage optimization, we've heard a lot on that topic, dosage optimization, but now I'm going to present a related topic, regimen optimization in breast cancer clinical trials. These are my disclosures. And this is an outline of my presentation. I will first describe what I mean by the concept of regimen optimization. I will then bring this issue more into focus by presenting a case example of pembrolizumab. Then I will go through some regulatory considerations for regimen optimization before concluding. And just to highlight that this talk is meant to be a short introduction with more discussion during the panel and Q&A. So what do I mean by regimen optimization? So far, we've heard about dosage optimization, and dosage refers to the dose or the amount of the drug as well as its schedule. However, beyond the dose and schedule, there are other components of a treatment regimen. For example, a trial might contain a multi-phase regimen with a neoadjuvant and adjuvant component, which includes, So for example, a trial might contain a multi-phase regimen with a neoadjuvant and adjuvant component. The relative contribution of each phase might not be established by that trial. However, the regimen will lead to an improvement in long-term outcomes. In that situation in the past, FDA has granted approval to an entire regimen. However, the consequences of this are that patients may potentially be overtreated and may experience avoidable toxicity. I will now bring this more into focus by talking about a very well-known trial in breast cancer, KEYNOTE-522, which evaluated pembrolizumab for neoadjuvant and adjuvant treatment for patients with high-risk early-stage triple-negative breast cancer. So the design of KEYNOTE-522, I think, is very well known to this audience. I will just go through it at a very high level. So this trial enrolled over 1,100 patients with early-stage triple-negative breast cancer, which was defined as high risk according to the trial eligibility criteria. Patients were randomized 2-to-1 to receive pembrolizumab or placebo in combination with neoadjuvant chemotherapy and then continued as adjuvant treatment following surgery. Some things to note about this trial design is that the patients continued with their assigned adjuvant treatment regardless of pathologic complete response information or PCR information available at time of surgery. Some other things to note, the primary endpoints were PCR rate and event-free survival, or EFS, and overall survival, or OS, was a key secondary endpoint. The PCR endpoint reflects the neoadjuvant portion of treatment only, and the EFS and OS endpoints reflect the entire neoadjuvant and adjuvant regimen. However, the relative contribution of each portion of the regimen is not established by assessing EFS and OS. This trial met its PCR endpoint, and at that time, EFS and OS data were immature. At that time, FDA discussed the results at an Oncology Drugs Advisory Committee, and the committee voted to await more mature EFS and OS data. Later, the EFS and OS information available on this slide became available. And as you can see, the trial met its EFS endpoint with a hazard ratio of 0.63 and the 95% confidence intervals shown, which favored the pembrolizumab arm. At that time, the overall survival also showed a beneficial trend towards pembrolizumab with a hazard ratio of 0.72 and the 95% confidence interval shown. However, OS was still a bit immature with 45% information fraction. Just to note, I do recognize that more updated overall survival information is now available from this trial. Just with the case example, I'm putting you into the shoes of FDA and the information that was available at the time of this approval. Then just to note that despite the EFS and OS information, which is positive that I showed you on the prior slide, this very important question is unanswered by the trial design. What is the relevant contribution of neoadjuvant and adjuvant pembrolizumab on the EFS and OS endpoints? And so you can see that this question is unanswerable due to this trial design and would in fact be unanswerable in any two-arm design. I also want to emphasize that even with a statistically significant overall survival improvement, that still does not establish that both the neoadjuvant and adjuvant portions, their relative contributions, or even that both of those portions are needed. I also want to review safety at just a very high level. So most of the toxicity that pembrolizumab adds to standard of care treatment is in the form of immune-mediated adverse reactions, which are shown on this slide. And although most of the immune-mediated adverse events occur in the neoadjuvant portion of treatment, patients can still experience immune-mediated toxicity during adjuvant treatment. And additionally, continuing to receive an intravenous adjuvant treatment necessitates more visits to a healthcare setting than if somebody were done with treatment. And I say all of that to just point out that the adjuvant portion of this regimen is not without added toxicity and added burden. In conclusion, FDA granted approval to this entire pembrolizumab regimen, including the neoadjuvant and adjuvant portions, for treatment of patients with high-risk early-stage triple-negative breast cancer. Even at the time of the approval, we noted that based on the trial design, it was unclear the contributions of the neoadjuvant and adjuvant portions of treatment. But further context on that, this regimen had shown an improvement on a long-term outcome in a disease setting where there was high unmet medical need. And in fact, there had been no new FDA approvals in this disease setting in several decades at the time of this approval. We do recognize, though, that there are important implications for clinical care and also future clinical trials. For example, patients might be potentially overtreated by receiving both neoadjuvant and adjuvant treatment. And additionally, patients receive very important information in terms of their individual prognosis in the form of PCR at time of surgery. And they may have questions about the adjuvant portion of their treatment, which these trial results don't help answer. And then additionally, of course, there's an ongoing clinical trial looking at whether patients who experience a PCR after neoadjuvant treatment need the adjuvant portion of pembrolizumab. Stepping out a bit, I want to also emphasize that a trial like this is not necessarily a one-off. We see in other disease settings that regimens are being proposed with neoadjuvant and adjuvant components, and additionally, in the future, we might see trials like this in breast cancer. And so I want to present, it's very important to FDA that these types of questions about whether all phases of treatment are answered up front, rather than all of us having these questions when the trials become available or when the drug is approved. And so I want to walk through some options for how to address these questions during the trial design phase, and I'll do that in the following slides. These examples all include a hypothetical experimental drug X. And so what might be some of the options for evaluating the contribution of the regimen? So one option could be sequential trials. So for example, trial 1 could add drug X for neoadjuvant treatment onto standard of care. Assuming that trial 1 is successful, in trial 2, then all patients would get drug X as part of their neoadjuvant treatment, but trial 2 would be evaluating what is adding drug X onto the adjuvant portion of treatment and what is the benefit of that. Another option could be a trial with more than 2 arms. I pointed out earlier that this type of contribution of phase question is unanswerable in 2-arm designs. So another option could be something like this. Arm 1 would include drug X added in the neoadjuvant and adjuvant settings. Arm 2 would include drug X added in the neoadjuvant setting only, and then Arm 3 would be standard of care. Comparing Arm 2 to Arm 3 could allow you to understand the contribution of drug X when added into the neoadjuvant setting, and then comparing Arm 1 to Arm 2 could allow you to understand the contribution of drug X then added into the adjuvant setting. Finally, and I won't walk through every arm on this slide, another option for optimization could be a trial that includes re-randomization. So I know there are a lot of arms on this slide, but I do want to point out that something like this, which includes re-randomization based on PCR information, takes PCR information into account, which is different than the examples I presented on the preceding slides, where patients continue with their adjuvant treatment regardless of PCR results. Before closing, I also want to highlight that although I have talked about regimen optimization as pertains to neoadjuvant and adjuvant treatment, there are other very important components of the regimen and important regimen optimization considerations. One of those considerations is duration of therapy, perhaps duration of therapy in an adjuvant setting. And you can see some of the questions that there might be about appropriate duration of treatment of experimental drug X. And some of these questions could be answered with sequential trials, with longer and longer periods of receiving drug X, as it has been done in several, of course, breast cancer settings in the past. Or it could be 3 or 4-arm trials where there are multiple arms of different durations of the experimental treatment being compared to standard of care. So in conclusion, regimen considerations are an important component of treatment optimization. Thoughtful trial design can support development of regimens that maximize benefit and minimize toxicity to patients. And there are ongoing efforts by FDA and multiple stakeholders to achieve regimen optimization. FDA held a public workshop in collaboration with AACR in spring 2024 on this concept of regimen optimization. Again, this talk is just a teaser on this topic, but if you're interested, please refer to all of the materials that are available online. This was a full-day workshop that discussed a lot of nuances of regimen optimization. And finally, I would like to thank the conference organizers for the opportunity to present on dosage and regimen optimization, which really feels very strongly that this is essential to patient-centric drug development. I'd also like to thank all of my colleagues at FDA who had input on my presentation. And finally, I would like to thank all of the patients, their loved ones, and all of the patient advocates who have talked to me over the years and talked to my colleagues at FDA over the years about the importance of this topic on their lives. Thanks to you all for your attention.
Hello. I too am very invested in this topic. I've been living with metastatic breast cancer for almost 12 years now. Thanks. I'm lucky. That's it. So I'm here to bring the theory of what we as patients experience on a day-to-day level. Really, all of what you've heard sounds good and true, but this is what we live with. Here are my disclosures. So cancer drug dosing, you've been hearing that more is not always better. Historically, it's been determined using these dose escalation studies where they increase the dose until they find something that patients can't tolerate, and then they go down and say, okay, this is what we're gonna move forward with. But this is really based on the idea that more toxicity is going to bring more efficacy. And that, as you heard from Pat and from Stacy, is often not applicable to these new treatments. And it also does not at all consider the long-term side effects that we as patients deal with. And that's especially true for those of us living with metastatic breast cancer, because if we're lucky, we're gonna be on one of these drugs for many years. So we did a survey of patients living with metastatic breast cancer. We had over 1,200 respondents, and we asked people about toxicity and what they experience when they are in treatment. We found that 86% of the patients experienced at least one, what they considered bad treatment-related side effects. Out of those, 20% had to visit the hospital because of a side effect to their cancer drug. And 43% missed the treatment because that drug was so toxic. So they couldn't get the drug because the drug is too toxic. And then we also asked about dose reductions, and we found that 83% of the patients that did have a reduction in their dose felt better after that. So again, what good is a treatment that you can't take? So the Patient-Centered Dosing Initiative were a patient-led initiative that was founded by Anne Lozier, who lived with metastatic breast cancer for over 10 years. Anne had this idea and really went after making change that patients with metastatic breast cancer are routinely treated with the highest tolerable dose. But MBC is not curable, but it can be treated, and often for years, sometimes even decades. We don't want to just live long, we also want to live well. And there's evidence that suggests that some of these drugs that allow lower doses can be as effective as the recommended starting dose, which is, most of the cases, it's the maximum tolerated dose. And then anecdotally, we've also seen several of these long-term survivors are on reduced doses for one reason or another, but that has played a role in how they can tolerate these drugs over long periods of time. So here's the Patient-Centered Dosing Initiative. We are 7 patients living with metastatic breast cancer, and we have an advisory board of oncologists who have been very helpful in guiding our thinking and designing our surveys and writing papers. So we know that this is how to get the attention of this audience is giving talks and writing papers. So on the top right corner, Anne gave an oral presentation at ASCO in 2021. We've presented posters. We published both the patient survey, and then we also did an oncologist survey, and we have published those. And then this also got the attention of the media. So there have been several articles in the lay media about how there is change happening about how cancer drugs are dosed. And this idea really gained traction with the FDA, as you've seen. So Atiq Rahman said at a Friends of Cancer research meeting that it's loud and clear from our patients that the drugs are too toxic. Patients deserve a more tolerable dose. And then in 2021, in November, in the fall, Mirat and Atiq and Rick and a team at FDA published a New England Journal of Medicine paper on when less is more. So as you've seen, real change is happening. Project Optimus was launched around that same time. And we are seeing more talk about post-marketing requirements and having to go back at some of these drugs that we find are too toxic for the patients. And then in the clinic, we really, as the patient-centered dosing initiative, we really encourage physician-patient discussions about the right dose for the right patient. So we want patients to be getting the right medicine and for them to be getting the drug that is, the dose that is right for them. Everyone has very different circumstances, and the discussion of dosing should be happening. It's a variable that should be addressed. We know that these approved drugs, sometimes it's really hard for it to change, for these dosages to change. It will take time for this to, for MTD to change and for new drug development trials to actually happen. And we have flyers, one for patients and one for oncologists, that are really designed to guide a conversation between patients and their healthcare providers. Sometimes as patients, we're afraid to bring up our side effects, and we're afraid that our doctor is going to suggest moving to another therapy if we can't tolerate the side effects. And we don't want to, because we only have a limited number of therapies. So here's a flyer, and we will have copies at the front that we're happy to share with you. There's also a flyer on for healthcare providers that gives some of the information from the survey, and we have those as well. So I hope I convinced you that there are many benefits to patients. So if there's less toxicity, then there's less of a need for emergency care. There's fewer missed treatments. We will remain on the treatment for longer, and it will work longer. We'll be able to take advantage of the full complement of treatments, and we will experience a better quality of life. And then maybe we'll even live longer. Oncologists clearly want the best for us, and Eric Weiner said, just because you have cancer and are on treatment, it doesn't mean you have to be miserable. We really don't want to be miserable. We believe that payers have a great benefit from this, so by paying fewer specialist visits, some of these emergency care visits, these additional medications, less absenteeism from work. And industry, we believe, the pharmaceutical industry, we believe, has great benefits from this as well, because if patients are taking the right dose, then there will be fewer post-marketing requirements for these drugs later on. Patients are going to stay on these drugs if they're working, and they're going to be able to tolerate them at better levels. And then in the real world, same, patients will be able to continue taking these medications for a much longer period of times. So I hope I convinced you that it's a win for everyone. We have a couple of surveys coming next year in 2025. We will look at ADCs specifically and dose reductions in those. And then we are also going to make an updated version of our patient survey, and that will be released in the new year. So check out our website, therightdose.org. Thank you.
Thank you so much to all of our speakers. I'm grateful to all of you who have submitted questions online. Reminder that also please feel free to go up to the microphones and ask us questions live. Just a housekeeping note, I realize we are not going to get to every single question. There is so much interest and engagement. So we will end the session promptly at 3:45, but a few of us will be available afterwards if you feel like you haven't gotten your question answered, and you can come up and chat with us. As people are getting ready with their questions, I wanted to invite my colleague Mallory Fierro to comment a little bit. A common question we get is, why does FDA care about randomization for dosage optimization trials? And do I have to prove that one dose is statistically superior to another dosage? So I'll invite Mallory to comment on that, and then we'll take some questions.
Sure. Randomization is a really important tool in any trial where we're trying to select the optimal dose. That's because we can compare between randomized treatment arms in terms of efficacy and safety. By doing so, we're ensuring that patient baseline characteristics are similar between the arms so that if there are any differences that we see between the dosages, it's due to the differences in dosages as opposed to any differences between the two cohorts. In terms of sample size, we do not require formal powering of efficacy endpoints in dose optimization trials. Really, what we're looking for is a large enough sample size where we can have sufficient reliability in being able to estimate our efficacy endpoints as well as for safety. The last comment that I also wanted to make was that we do value and take into account patient-reported outcome or PRO data into these trials, and particularly for randomized trials. And ultimately, we evaluate the totality of the data when we're looking at these trials. Thank you.
Thanks, Mallory. I think I'll start with the microphone to my right, and then we'll switch off.
Daniela Presco, medical oncologist, UK. I just want to ask the panel, in the phase 1 clinical trial design, do they still use the modified Fibonacci dose escalation? Because that might reflect why we experience problems with the narrow therapeutic index. Is there any other mathematical model which is used in the phase 1 clinical design? Because as we know, this modified Fibonacci dose escalation reflects heterogeneous variation of a genuine sequence, which doesn't correspond to a constant number of the drug at the high doses.
Thanks very much for the question. And if I understand correctly, you're asking about kind of this best options for steps between dosages in a phase 1 trial. Pat, maybe I'll start with you, and then I'll invite others to comment.
Well, first of all, we rely heavily on the biostatistician, and I think she wanted to comment, and then maybe I can afterwards.
Yeah, sure. There are a variety of different additional approaches that could be used for dose selection. So, for example, we can have any Bayesian approaches, Boyne designs, MTPI, things like that. From an FDA perspective, we don't mandate any particular trial design. Ultimately, what we want is to have an approach that has good properties and characteristics so that we can have enough information to be able to select the dose.
Right, thank you.
I would like to make a comment if nobody else on the panel would. I'd like to say too, though, part of this dosage optimization conversation is a little bit about the goals of different phases of drug development. So regardless of 3+3 or some of the newer Bayesian approaches, it's also about reframing the goals and moving away from the goal being solely to identify the maximum tolerated dose to more of a holistic assessment of the benefit-risk trade-offs among various dosages of the experimental drug.
And, we do sometimes modify the doses, either through an amendment or the way we originally write the trial design to look at intermediate doses so that we can hopefully get it right.
Thanks. I think we'll go to microphone on my left.
Thank you. Paul Kortz from Lisbon, Portugal. I'm a medical oncologist. I would like to look at the pembrolizumab case in a different manner. So initially, the trials were per milligram per kilogram, and then the pharmacodynamics went up to have a fixed dose. Probably the dose is more than most of the patients need, and in pharmacoeconomics as well. Canadians are going back to the milligrams per kilo. What's your thoughts about it?
So it depends on what drug type we're actually looking at, on whether or not what the approach is to figuring out the dose. So some of our doses are still based on milligrams per kilogram, and we've seen that quite commonly for like the ADCs that still have somewhat of a more narrow therapeutic range because the payload's typically cytotoxic. For oral drugs, it's just easier to have a flat dose, and usually there's a wider therapeutic range, and it just makes it easier to dose. With some of the other drugs as well, with some of the biologics, we also see, a fairly wide range, and it's allowed us to go from a mg per kg dose to a flat dose. I will tell you from the clin pharm perspective, we do look at weight as a factor, and I think there was actually a recent approval that we've actually kept the mg per kg dose at a maximum dose because we knew there was issues with toxicities in certain weight categories. And then there certainly has been some of the immunotherapies that there's been weight cutoffs for dosing. So I think weight and body size is a factor that we look at within the analysis and trying to figure out the best dosing approach, and we try and look at that early with the companies or sponsors early on to figure that out. But there's a reason for it. It has to do with how the dose response or exposure response relationship curves look.
Nasir?
Thank you, Pat. It's actually a question to you. Before you move to the phase 2 and determine the recommended dose, what is the acceptable figure for treatment discontinuation due to toxicity?
Can you repeat the question?
Treatment discontinuation. What is the acceptable figure?
Treatment discontinuation because of toxicity?
Yep.
Well, if we're getting, number one, it depends on the patient and their underlying comorbidities. And number two, it depends on the toxicity that they're having. But for the most part, we really, especially within the first few cycles, if we have treatment discontinuation because of toxicity, we want to either expand below that dose, understand that dose more, because when we only have, like 6 patients, and if one of them is discontinuing because of toxicity, we're going to want to expand, and some of the other patients are having some issues, we're going to want to explore those doses to a greater extent. We don't want, occasionally you'll get a patient that discontinues, but we don't, I personally, in the years that I've been doing this, don't like that, because if I have one or more patients that has to discontinue, I've probably hit the dose wrong, and even if they have to discontinue in cycle 3 or 4, it still makes me think I'm still too high. You got to look at, I don't know that AI will take over our jobs in phase 1, maybe it will one day, but I think we have to look at the total picture of what's going on. Because I don't like dose discontinuations and then moving that dose forward. It's somewhat uncomfortable for me. I don't know if you
I can make a comment too. I think some of these questions are really challenging to answer when there isn't a comparison. And so Mallory was talking a little bit about, what's the importance of randomization, but the other element of those trials is that 2 or more dosages are being compared. So you can understand, for example, dose reductions are 30%. Can I improve on that by still be in kind of a therapeutic dose range, or is that kind of the dose reduction frequency I'm going to look at? And it's very hard to know the answer to that question if you're looking at one drug dosage in isolation.
Yeah, no, I agree.
We have a lot of questions online, so I think we'll go to this microphone on my left, and then I'll answer one or two, or I'll ask one or two of the online questions, and then we'll pick up with the live mics again. Go ahead, please.
Judy Ann Chapman. A question about the beautiful idea of getting biomarkers right from the beginning. I'm hoping that when you're talking about prediction, you're not talking about taking that beautiful new biomarker and categorizing it, rather than keeping it continuous. Because I think that has led to a lot of problems in practice. When you right up front categorize something, and you got that wrong too. The dose may be wrong, but the biomarker cut point is likely wrong.
Oh, no, I agree. You have to, that's why we have to study it. And many times in early phase trials, even if we do have biomarkers, they typically are the exploratory objectives because we probably haven't validated it. It's probably definitely not in CLIA, it's not CLIA certified, which I know is not an FDA mandate, but is a clinical mandate. So most of the time when we're developing biomarkers early in phase 1 trials, unless it's beyond first in human, first in class, those biomarkers, and often we have a panel of biomarkers, depending on the drug class, will be exploratory so that we can understand their relevance.
Thank you.
Yeah.
We have a few online questions on the topic of PROs, so I'm just going to read one of them and just know it's representative of a few people's questions. So the question we got is this the time to embed the overall concept of dose optimization around patient-centric outcomes, including PRO and overall quality of life in trial designs? Maybe Mallory, I'll start with you.
Sorry, is it the time to what?
How do you think about can you broaden a little bit what you said about incorporation of patient-reported outcomes into dosage optimization?
Yeah, sure. For patient-reported outcomes, it adds such valuable information to understand how patients are feeling and functioning while on treatment. This is certainly an area that's quite new and that's rapidly evolving. In terms of the symptoms, if you're considering a randomized trial for dose selection, it's important to carefully select symptoms that are related or most applicable to the disease setting in the population of interest. FDA does have a guidance on core patient-reported outcomes in cancer trials that you can refer to that has some recommendations on symptoms to select, but this is very important data.
Thanks, Mallory. Julia, would you like to comment as well?
Sure. So we know that clinicians may underreport patients' adverse events. So in this phase where determining the dose will be moved forward, and that's what all patients will eventually take of this drug, I can't think of a more important time to be asking patients directly how they feel on the drug.
Thanks.
Can I make a comment as an early clinical trialist?
Yes.
So one of the concerns I have is that in the early phase clinical trial category of trial phases, especially phase 1, I don't know that we collect enough PRO data. And I don't know yet, has there been an example that we can clearly state a dose was defined in early phase based on PRO outcomes?
Thanks. So I'll make some comments from my perspective as an oncologist and then also as a regulator. So I think one of the ways in which PRO information can be particularly helpful for dosage optimization is those low-grade symptomatic toxicities, which people tell us are extremely bothersome, especially over long periods of time. Things like diarrhea, things like visual impairment, which may never be a grade 3 or grade 4, but just are extremely bothersome and affect the experience with that drug, affect dosage interruptions, dose reductions, and even discontinuation of the drug. I think PRO information can be very important additional safety information to have during early phase development, including if you're considering multiple dosages and trying to decide which one to take forward. And so, yeah, just like we were saying, I don't think, just like we shouldn't be using only high-grade toxicities to define the dosage, I think it's hard to say that we will get to the right dose using only PRO information, but as a component of the totality of data, I think it can be extremely important to support dosage optimization. I think we'll go to this microphone on my left. Please go ahead.
Hi, it's Stephen Vogel from New York. For Dr. Shah, my ears went up when you said you want to optimize the duration of therapy. And I just wanted to raise an issue about when you should randomize to further therapy versus not, and how you should balance the patients being randomized to do that. Ideally, you have to, if you have a toxic drug, you're going to have if the patient is still willing to take more, and the patient hasn't stopped it already after, let's say, a year, you need to take the experience of the first year of that patient, bad or no problems at all. Those are different patients, and the quality of their lives will be different from a second year. A lot of patients, if they're very sick for the first year, will just walk out of the clinic and say, you're not giving me this anymore. So I think if you allow the study to go from a randomization before the patient has ever gotten any drug, you get very poor information, and because of non-compliance, early cessation, the arms will look more the same than they actually are, and you could miss a benefit from continuing the drug.
Thanks very much. And can I just say it's an honor to have received a question from you at this conference. I think it's been my dream since I was a fellow. So thanks very much, and I think I'm going to take my typical regulator prerogative and say it depends. Totally understand your point, and I think a little bit matters on what we're thinking about, what duration of treatment we're looking at. So, something like 5 years, 10 years, yeah, I think it would be very hard to do that randomized trial up front. We really need information from that first period of treatment, and scaling that back, if the question were more like, 4 months or 8 months, or things that are smaller amounts of time. It might be a little bit. It might be a little bit more feasible to conduct that multiple arm trial up front and have that information in an expeditious way. But I totally hear your point.
This was done in colon cancer with positive nodes. All right, 3 months versus 6 months. The randomization was prior to entry. And about 15 or 16% of the patients never got to 3 months. There wasn't much of a difference, but they diluted the difference by including those patients who never could get 6 months because they never got 3.
Yeah, thanks very much for making that point. Pat, did you want to comment? No? Okay.
No.
Okay.
I'm still perplexed with the question, but thank you. Thank you for the opportunity, but I'll decline.
Let's go to this microphone on my right, then we'll take an online question before going back to the mics.
Thanks so much. My question is how to do this research. I'm representative for one cooperative group in Spain, Solti, and every time that we try to offer any type of de-escalation trial, optimization trial, biomarker-driven trial, is no and no and no. How to get sponsors, how to get money, how to get resources, how to keep this academic research into real practice. As all our clinicians, we would love to put our patients into all these ideal clinical trials. All of us do agree with your ideas about how to do that.
Thanks very much for the question. Julia, would you like to comment?
Well, you're the one that has the stick. So I think they are coming around. I mean, there's really no choice. But yes, I hear that all the time too. They have to be made.
I do think that it is a lot more feasible to try and get the trial right in the pre-approval setting for a lot of reasons. Stacy alluded to, some of the challenges of getting trials complete and information in the post-market setting. And I think that's due to a myriad of factors, but part of it is, especially in the US, where being on a clinical trial usually involves a lot of travel to a clinical trial site. I feel like it's very understandable that people might not want to enroll onto a clinical trial for a drug that's already approved and that they could get much closer to home. So I think that's just one of the feasibility challenges in the post-market setting. I think it is a lot easier to do the right trial early and address some of these dosage optimization questions early. Thanks very much for the question.
But I think your question was more focused on the clinicians want to do the right trial. After they've seen the results of a phase 3 and a drug get approved, they realize that there's questions that haven't been answered, and they want to design a trial to answer those questions like, is there a relevant biomarker? Do we have, whatever? But what I think you're asking is, the challenge is, how do we get those trials funded? And how do we get them through? Because we need drug and we need money. I think that's what your question was.
Yes.
Yeah, and trust me, it's becoming a greater challenge. and there's usually a cap to what many companies can give you for those. If they're going to accept the IIT, there may be a cap to the funds, resources that they're going to give you. And serial biopsies are extremely expensive. That is a real challenge. How do you do those trials? And what are the requirements to having that data?
Thanks so much, Pat. I'm going to take one online question and then perhaps this last microphone question before we switch topics. So we've gotten a few online questions on the topic of optimized dosages in different therapeutic settings. So for example, one question is, can you extrapolate optimal dose in one therapeutic area to another? I think I'll start with Stacy and then see who else wants to comment.
So again, I'll be the typical regulator and say it depends. So there's a lot of different circumstances on how we develop drugs. Some of the first-in-human trials have all comers, solid tumors, hematologic malignancies. We can start to see response and activity and safety data, and that can help inform the dosages across. Other trials come in and they're very targeted for a specific marker, for a specific solid tumor, and then therefore all the development has been in that disease. So I think looking at non-clinical data, looking at what clinical data you have in front of us, there's a lot of model-informed approaches that we can use to kind of figure out how to move the dose that we know and understand into the next population. And it may not just be different cancers. We're also talking about moving from, late-line metastatic cancer to front-line metastatic cancer, or even into the adjuvant setting. So all of these model-based approaches, non-clinical data, the emerging clinical data, all can be used together to try and figure out how much clinical data, how much new studies do we need to do to really answer that question of figuring out the dose in the next population. We're not expecting dosage optimization in every single setting and every single disease state because the resources just aren't there. They're not there from the patient perspective. They're not there from a lot of different perspectives. But I think the general idea is that we want to just try and maximize the use of all the data that we have in front of us to make an informed decision on what the best approach is to optimize the dose in the next setting.
And honestly, I think that a lot of it depends on the biology of the drug and the biology of the disease. And so predicated on what tumor type you may be targeting relative to given drug X, what are the mechanisms of resistance, the more common mechanisms of resistance against this tumor relative to another tumor may in part predicate dose. And if you're looking at the biology with serial biopsies, you may identify that some tumors may need more drug to maximize inhibition or alter the tumor microenvironment than other tumors with the same drug requiring maybe different doses. I think that we're starting to realize how important biology of disease and biology of drug is and how to best marry that.
So there's one other point I want to add to that too, is it also depends on the patient's reserves. If you have a metastatic breast cancer patient who's already been treated on their second or third drug, they may not be able to manage some of the toxicities that are coming with the next drug because their body has already taken multiple hits. If you have somebody who's adjuvant cancer, the question is you don't have tumor on board. Like that's the goal, like to keep it at bay. And so the question is what dose do we need? And those patients may be a little bit healthier, may be able to tolerate a slightly higher dose, and you're in a curative setting. So I think all of those things need to take in consideration of like where we're going forward. And we do have examples of some drugs, especially in hematologic malignancies, where the biology of the disease drove differences in dose, and we've seen that quite a few times in hematologic malignancies. We've also seen we've seen differences in solid tumors between, with some of the gastric cancers, we've seen some differences in dose needed with some of the drugs.
Thanks. Michael, did you want to make a comment? I saw you leaning into the microphone.
So the drug development is being accelerated, and we realize that we need to spend more time early on and use biomarkers, trying to understand early on what's the best dose. And to the comment about the regimen optimization, try to do that also early on before approval, as you were saying, because after that, it's really difficult to have funding, and it's really difficult. It may be too late given all the resources that have been spent. So it's a plea to work more before we jump on the phase 3 trial.
Thank you. Last question on this topic, please go ahead.
Okay. Michelle Tregear. I'm with the National Breast Cancer Coalition, patient advocate. First, I wanted just to applaud the patient-centered dosing initiative, an advocate-led initiative for really driving change in this space. I think patient dosing is a really important thing, and I'm glad the FDA has taken it on. And I was really intrigued to hear about the regimen optimization. So I guess my question, especially giving the example that you cited, Keynote 522, has the FDA given any thought about standard of care optimization for control arm participants? Because I think some would agree that perhaps the subset of patients who didn't receive a PCR in that trial in the control arm might not have had the best standard of care. And and I think it's really important as we're in a space where new drugs are coming to the market all the time. How can we protect patients who are in those clinical trials, especially if they're on the control arm?
Thanks very much. So I think a couple of questions which are related within the question you asked. So a little bit on the control arm. So generally when we look at a trial and design phase, the control arm has to be something that reflects standard of care treatment, but we do recognize that there are different options for standard of care treatment. A related question I wanted to answer, sometimes people ask, if I'm putting a new drug in combination with an old drug, and the old drug is approved and it has an approved dose, but there's reason to believe that that approved dosage, might be based on the maximum tolerated dose, is not optimized, do I then need to use it in this new combination? And the answer to that is no. You can evaluate a new dosage of that approved drug in combination, and that might overall lead to a regimen that's more tolerable than if you were to go forward with the current label dosage and put it with a new drug. Does anybody else on the panel have comments on this? And thanks very much for the question.
So the next session, which is the last session and the last speaker, is Dr. Michael Ignatiadis. Sorry about that, Mike. And he's going to be talking about the lack of crossover in clinical trial design. So we're shifting away from Project Optimus here. Mike?
Thank you. I would like to thank the organizers for the opportunity to discuss about this important topic. So these are my disclosures. So what is crossover? We all know in the context of randomized trials, crossover is when patients in the control arm at the time of recurrence or at the time of disease progression receive the experimental arm. So what I will discuss a crossover that is specified in the protocol, these cases. I will not discuss cases with crossover that is not specified in the protocol but occurs after unblinding in the case of a positive trial. So first of all, let's see, is crossover common? So these are data from the US. From 2000, in the last 30 years up to 2020. And so basically what they have done, these investigators, so they have looked in the different trials in the last 30 years, and they found out that out of 459 trials, only 35 had crossover, so around less than 8%. And now if you look at these 35 trials, now 5 trials had designed, crossover design, so it was incorporated in the design. It means that all patients did crossover. Basically it's trials that are testing sequencing, regimen 1 followed by regimen 2 versus regimen 2 followed by regimen 1. And then there are what we call crossover allowance trials in which crossover is allowed, but it's not mandated. So let's see in the 30 trials that allowed crossover. What happened? So the median number of patients that did crossover range from 2 to 88%, and so the median was 57%. So this was the median number of patients in these trials that did the crossover. So crossover seems to be more appealing for patients. It may add in the recruitment, although this is controversial. Some people think it cannot add in recruitment. It might be ethical in specific settings. On the other hand, it can confound important endpoints such as overall survival. So let's see two clinical scenarios with examples. So first scenario, the experimental drug is standard of care in later line. And second scenario, the efficacy of the drug is not established in any lines of treatment. And let's start with the first scenario. So the experimental drug is standard of care in later line. And I will quote this from JCO, from Sheffer and colleagues, that patients who have chosen to take on the risks and burdens of clinical trial participation to help generate knowledge in support of medical programs are owed appropriate standard of care post-progression. So if there is effective standard of care post-progression, if the experimental drug is standard of care in later lines, they should receive it. So let's see an example from prostate. So this was published in the New England in 7 years ago. The abiraterone in metastatic castration-sensitive prostate cancer. As you can see, there is a nice overall survival benefit from abiraterone. However, abiraterone was available at the time the trial was conducted as standard of care in later line. Patients randomized to placebo, a significant proportion of those patients did not receive abiraterone, as crossover was not initially allowed. And so the overall survival benefit that one can see here, we don't know whether it's driven by the fact that patients in the placebo arm, or at least partly driven, did not receive abiraterone as standard of care when they have progressed. And so in a letter to the editor for this specific trial, De Bono and colleagues said the drugs used in the placebo group were inconsistent with the prevailing standard of care, and this has implications for the conclusions of the trial. So if we go to examples for breast cancer, in this commentary in Nature Reviews Clinical Oncology, this author asks, what is the percentage of women enrolled in the NATALY and in the Monarchy studies that receive standard of care CDK4/6 inhibitors at the time of relapse, especially in low middle income countries? So what is the percentage of patients in these two trials that did crossover, and how this is going to affect the interpretation of overall survival results when these are going to be reported. Now let's take another scenario. The second scenario, where the efficacy of the drug is not established at all, and see what is the effect of crossover in this situation. So this is again an example from prostate cancer, in which investigators were testing a new agent, Sipuleucel T, for castration-resistant prostate cancer. And of the 171 patients in the placebo group, so this was a placebo controlled trial, half of them received this new agent at progression, so did crossover. Interestingly, in this study, progression-free survival was not improved. However, overall survival was improved. And then there was an RQ report suggesting that the trial was positive because the crossover that happened in this trial harmed the control group by delaying an alternate effective therapy, which was docetaxel. So basically they say this drug was not working, patients in the control group was allowed to do crossover. That's why you see the survival benefit. Another example in which crossover was not allowed, in example from breast cancer in the Cleopatra trial. In this case, the efficacy of pertuzumab in later lines was not known, crossover was not initially allowed, and the trial was positive for both progression-free and overall survival. So here, in this situation, the lack of crossover allowed investigators to demonstrate not only PFS improvement, but also improvement in overall survival. And so why this is important? Because in the ESMO magnitude of clinical benefit scale, in which it's a beautiful scale that can evaluate drugs that have been approved by FDA and EMEA, and helps to frame the appropriate use of limited public and personal resources to deliver cost-effective and affordable cancer care. And in this situation, the crossover and overall survival benefit may change the score of a given drug, and thus the prioritization of a given drug in healthcare of several countries. So in conclusion, crossover should be mandated or at least encouraged when the experimental drug is standard of care in later line. Of course, if cannot be mandated, it should be encouraged. And in this situation, information on post-progression treatment needs to be collected and reported. And this information is often missing, even from high-impact publications in New England Journal of Medicine. Now, on the other hand, crossover should be avoided, as you have seen, when the efficacy of the drug is not established, because it can lead to spurious conclusions about the efficacy of the drug. So I think I will stop here, and I would like to thank my colleagues at the EORTC and statisticians there that I had useful discussions for this talk. Thank you, and happy to take questions.
Thank you very much, Mike. I don't have access to the questions online. Yes, to the left.
Yeah, thank you. I think that the FDA should require, even more than strongly encourage, crossover for many different clinical trials. And I think in particular, the instance of first-line therapy with a combination of endocrine therapy and a CDK4/6 inhibitor, because the combination got embedded before the results of a crossover trial called SONIA showed that there is not a long-term benefit to upfront therapy with the CDK4/6 inhibitor for the patient population overall. But it's been very difficult for the SONIA results to get much headway against the previous data.
Thanks very much for the question. Maybe Mallory, you could start by making some comments.
Yeah. From an FDA perspective, crossover and initiating subsequent therapy are very important parts of trial design where patients can get access to additional or subsequent therapy after progressing on the control arm. FDA does not, or we cannot mandate whether or not sponsors permit crossover in their trial design. There are some considerations. For example, allowing crossover can actually help prevent asymmetric dropout in an open label trial, such that if patients have knowledge about their treatment assignment and they were randomized to the control arm, if perhaps there's some understanding that the crossover is allowed, then patients may be more likely to stay on the trial, and we're able to have more reliable results for things like progression-free survival. Alternatively, crossover can potentially have bias in cases where an investigator might have perceived superiority of the investigational arm, where maybe the investigator is calling progressive disease earlier on the control arm compared to the treatment arm. So in that case, there can be concern about potential bias and therefore a higher percentage of patients that cross over. Ultimately, from an FDA perspective, we always look at the percentage of patients who cross over, the reasons for crossing over. In breast cancer trials, overall survival may not always be feasible to formally power. However, we always look at overall survival in these trials to ensure that there's no detriment. And if crossover is permitted in the trial, we wouldn't expect to see any potential detriment in overall survival in those cases.
Are there questions online, do you know?
Just one, which I'll read, and then perhaps Michael, Mallory, and anyone else who wants to comment can comment. So the question online is, when the experimental drug is standard of care in later line, the endpoint can still be overall survival because the question is about the best sequence to improve overall survival, question mark.
Yeah, so in this particular situation in which the experimental drug is standard of care in later line, I think, as we discussed, its crossover needs to be encouraged. First of all, for ethical reasons, for patient participation reasons, but also it's important for us to know when we're going to interpret the results in terms of overall survival. So imagine a study in which the crossover was allowed in only, happened in only 10% of the population, and in the control arm, and a situation in which it happened in 90%. So, of course, if there is an overall survival and virtually no patient in the control arm received the standard of care, then of course this overall survival benefit is actually driven by giving the drug or not in this patient. It's not by giving the drug in the adjuvant setting. So I think what it has been said is we cannot mandate, but we need to have this info so that we can interpret the results of a trial and we can interpret the benefit we see. So this is a very important info that we need to collect. So in that specific scenario, we need to collect information on the treatment post-progression. Sometimes, unfortunately, this is not the case. It's not being collected. And so we cannot have, and we cannot say, is this overall survival benefit really driven by giving the drug early on in the adjuvant setting, or it is there just because patients in the control arm in those countries that have been done didn't have access to this drug in later line and didn't receive it.
Oh, yeah.
Maybe I can add, from a statistical perspective, sequencing is very different from a randomized trial where we have drug X compared to a control arm. So if there is interest in understanding the impact of sequence, it's really important to pre-specify that type of thing in the trial design. But at the end of the day, if we are looking at, say, one investigational product versus a control, we do look at the percentage of patients who have subsequent therapy, including we have discussions about different regions, generalizability from the US population. These are very important as part of our benefit risk assessments for FDA.
Okay, I think time is here. I know that several of the people on the panel here will be available afterwards if you have any questions up front here. If not, I think this concludes the session, and we thank you very much for joining us this afternoon.